Are you ready to start with every day as a school day? I know that's stepping out of the normal pattern of our Discovery Matters podcast, but Conor, I cannot hold back. I learned something incredible.
Oh my God, if you learn something incredible, I want to know it too. Go on.
All right, so recently in late October, doctors at Norrland University Hospital in Umeå, way up north in Sweden,
in the cold depths of darkest winter. Yeah,
that's right. Tested something called magnetic camera-guided focused ultrasound to perform a 15-second procedure, non-invasive, on patients who have involuntary tremors.
Okay, so this is people who can't control the shakiness in their limbs.
That's right.
Or people who have Parkinson's or other tremor-inducing diseases. Is that right?
That's right. So it directs ultrasound at specific points in the brain and with heat permanently disables those areas that are causing tremors.
And why are you telling me right now, before we meet the excellent experts that we usually speak to on an episode of Discovery Matters?
I'm so glad you asked, because today's episode is about different ways of understanding Parkinson's better and perhaps diagnosing the condition earlier than ever.
Because early diagnosis means better treatment options, means better health outcomes, so tackling Parkinson's disease is what matters on today's episode of Discovery Matters, brought to you by Cytiva.
Say hello to Mariana Dichet, whose team discovered a new genetic mutation in two sisters in Oman. And that geographic and cultural note is important, as we will all learn later.
Okay, but first, tell us what made Mariana curious enough to be working on Parkinson's.
I would start by saying that SGIP1, which is the protein we looked at in our study, the protein came to us. So in the lab of Patrick Verstreken, where I did my research, we focus a lot into synapse and synaptic homeostasis in general, both from a physiological point of view and a pathophysiological perspective linked to Parkinson, like in this case, but other neurodegenerative diseases as well. So we've been already for many years involved in research on how different synaptic proteins are mutated in familiar forms of Parkinson's.
And how did the discovery of this particular protein come about?
Mariana described this as the protein coming to her. Pretty nice, isn't it?
I like that.
Yeah, it started with a collaboration between the lab of Patrick Verstreken and the Sultan Qaboos University Hospital in Muscat.
They, in the clinic, were treating two sisters affected by juvenile Parkinsonism, and they found out that they were carrying a homozygous mutation in SGIP1, a novel never found before. They reached out to us because of our previous research in synaptic protein mutation linked to Parkinson. We were immediately very excited about it because indeed this is what we were studying, but also because clinically the sister had very similar symptoms than patients that carry mutation in other synaptic proteins, which are synaptogenic and DNA-JC6, which we have previously studied in the lab. Very importantly, the SGIP1 was initially identified as an endothelin A binding protein, and other project I was carrying out was on endothelin A. We had extensive research done on this protein in the lab, so we were very intrigued by the link between all those proteins.
Oh, we do love an acronym, so what does this one mean?
SGIP1 is an acronym within an acronym, actually, because it's SH3GL2 interacting protein 1, SGIP1, which is the crowning of endothelin A interacting protein 1.
I'm going to pretend I know what that means. Could we just go back to what you mentioned earlier? This collaboration was with the Sultan Qaboos University Hospital in Muscat.
What happened with those recessive mutations, they become clinically apparent and manifest only when they are homozygous. So you need both parents to carry those mutations. And because this is very rare mutation, it's also rare that the parents are carrier. So the family, they were cousins. it happened that some of the members of the family were carrying it in homozygosity. And that's how those mutations became apparent, and that's how we found out about them. It's very bad for the family, but it's good for science because at least we have a way to study the pathophysiology of the disease, which in most cases has idiopathic origin, so it's a multifactorial disease. If we have a mutation, then we can easily model it in the lab and see which molecular pathway is affected and try to better understand what's going wrong in the brain of those people.
Quick definition here: homozygous, having two identical alleles of a particular gene or genes.
Right, now that this novel discovery has been made, what does that mean with respect to the direction of Parkinson's research or the diagnosis of Parkinson's?
Well, previous research has predominantly been diagnostic and concerned with the pulmonary loss in this disease. In the last few years, it's become more evident that synapses are important in understanding the disease.
Now the field is moving, not all of it, but a part of research field on Parkinson is really looking at synaptic physiology and specifically at the proteostatic mechanism, so how protein quality control is carried out and dealt with at synapses, which are very specific compartment of the neuron and very sensitive also due to their physiology. And this SGP1 is one extra protein that belongs to those pathway, that belongs to that location. What we have been doing in the lab was also to try to group those mutations, because there are more than 20 genes that are being found mutated, of which mutation have been linked to the development of Parkinson and Parkinsonism. They all appear to be grouped. Some proteins, some genes that are mutating encodes for protein that have a role in mitochondrial health, some other in the endolysosomal system or autophagy, so protein quality control. Knowing if somebody is carrying a mutation, maybe it helps already giving a direction to the treatment. We're not that far.
So what does all of this research mean if we're aiming to get to a place where maybe Parkinson's is either preventable or curable?
In this case of the family that we have in these studies, it's a bit of a complex symptomatology that they have. So juvenile Parkinsonism is not only giving the motor symptoms that are typical of Parkinson, but one of them has epilepsy and both of them are cognitive impairment since birth. So they also have a little bit of a developmental issue. So this unfortunately cannot be reverted back. But generally, Parkinson patient, age is the number one risk factor, let's say. Or even if it's a genetic cause, it doesn't occur this early, like in juvenile cases. So now we know that the disease progression at the molecular level starts more than 10 years before the motor symptoms appear. But right now, our clinical diagnosis and definitive diagnosis still comes with the motor symptoms.
So she's saying it's too late by the time we see it.
And it's very often the case with neurodegenerative disease. By the time the symptoms present themselves in a way that the patient goes to their healthcare provider and asks for a diagnosis, it is too late. So that's why the goal of these studies is to be able to identify these diseases very, very, very early.
The motor symptoms comes when the dopaminergic neurons die, and then it's too late. But here we're looking at synapses, and we have this idea now that synapses are affected earlier, and then there are mechanisms by which perhaps the neuron dies back from the synapse, or even before the degeneration of the neurons. The neurons are all connected. One neuron doesn't fire or doesn't release the right amount of neurotransmitter because it cannot do that anymore. The whole circuitry will be affected. And this necessarily has a consequence in the way we function as a whole body. We know that some of the early symptoms of Parkinson's disease are, for example, constipation, hyposmia, which means that we have difficulties in smelling and recognizing smells, or REM sleep disease, so you have disturbances in the sleep pattern. A lot of attention is being given also to identify markers that are easily detectable in a non-invasive way. because we can do PET scan with tracer to see that the synaptic density in the brain is reduced, and then we know it's already something wrong, but it's not that we can do a PET scan to everybody. It's not sustainable. So drawing some blood or urine and looking at it, it's much easier.
Let's dive into the big question then. What is the biggest unanswered mystery in Parkinson's research?
It's a matter of decoding the why behind the how.
I think it's still how all those mutation in different genes that seems to be unrelated, how all of this come together in one clinical manifestation. If we look at genetic and familiar causes, because as I said, the majority is still idiopathic or of unknown origin, more environmental exposure of pesticide or whatever toxin. So how can it be that we all have a single well-defined clinical manifestation of this? How come that certain neurons are more vulnerable, and indeed define better step by step the mechanism at synapses that are disrupted and how to target them really? Because right now we're still using symptomatic treatment. So this is a big thing. Given that the age of the population is increasing, the average age, we have more and more people suffering from Parkinson and degeneration in general. We need to find a better way to treat them.
Now, leading on from our conversation with Mariana, is it possible to diagnose Parkinson's disease early enough using biological samples, for example, such as blood that we have access to? And what are the approaches that we can use using biomarkers to detect and monitor disease progression before we start to see significant impact from the disease?
So Mariana did mention that a lot of attention goes toward identifying biomarkers that are easily detectable and non-invasive. But to talk more about this, we spoke to Dr. Michael Bartle, and his team has developed a blood test to diagnose Parkinson's disease 7 years before symptoms begin. 7 years.
Parkinson's disease is not a treatable disease, and so we need new pathways and biomarkers. So we come from the biomarker field, we work a lot with different approaches, we do a lot of proteome analysis just to see if are there differences comparing Parkinson's disease patients and healthy controls. Parkinson's disease is diagnosed with these typical symptoms, motor symptoms like bradykinesia, tremors, or have this rigidity of the muscles. And when we diagnose this disease based on these key features, then we know that most of the cells in this midbrain area are called the substantia nigra, which is the main area where you have these dopaminergic cells that are very important for the regulation of the movements. 70-80% of them are already gone. So when we diagnose the disease, we are too late from the point of view of protecting cells, of preventing the disease to occur, and so on. So we have the two main goals. One is diagnosing the disease and having markers that are correlated with the symptoms, and to do this very, very early.
Before we get into the nuts and bolts of the study, I asked Michael about his personal connection to Parkinson's disease.
My grandmother had Parkinson's disease, so before I got a doctor, I was already confronted with this. So this was not new for me. I knew how it could look like, and I knew that they were very limited in the options that we had, and it worsened, and there was progression. And then I got connected with Britt Mollenhauer, Professor Britt Mollenhauer, who's my supervisor. She is the leading physician of this clinic, and she's very keen and very experienced in the research. So this came together. When I saw some patients and saw that this is not a rare disease, and these patients are really affected by the disease, there's a need, and when things come together, then you see you really should do something about this.
There's a huge shift in how we approach Parkinson's. And imagine if by using advanced biomarkers and predictive diagnostics, we could identify Parkinson's much earlier, like you were saying, Conor. And those critical early stages could help patients before symptoms show up.
So early detection and new clinical research, obviously critical. How do they improve patient care and outcomes?
We should always have a look on the patient and their needs and what we can do for them. If we could give these predictive results to patients, the only consequence would be that he knows about it, can plan maybe and so on. But the patients that would get these test results would for sure be the ideal participants in a clinical trial where you want to assess the efficacy of a potential neuropreventive, neuroprotective trial. You need biomarkers that are directly correlated to the symptoms. If there are new drugs, then it would be very important for patients to get this diagnosis very early. Even if there are no drugs available, it would be an option to give this information early and to avoid maybe a long journey through different doctors, maybe also expensive journey, maybe or also uncomfortable journey, a lot of diagnostics going on because it's not so easy to diagnose Parkinson's disease, but maybe also tells something about the potential cause of the disease. We know that there are different phenotypes of the disease. Some of them progress very fast, some slow. So that's also a very important aspect of our research.
What did Michael and his team use to identify and validate the biomarkers for Parkinson's then?
One important aspect was their use of proteome data and how machine learning was applied to distinguish between healthy individuals and those at high risk. Not the first time we're talking about machine learning in science on this podcast, is it?
We had blood samples from Parkinson's disease patients and from health, sex, and also educational matched healthy controls, and just to see are there differences, are there significantly differently expressed proteins and so on. We got from this more than 1,200 proteins that are very different. That's a huge number. We use mass spectrometry to just segregate molecules and to analyze different fluids. Then we put together these differently expressed proteins and we validated them. We built up a panel consisting of 20 proteins that were very consistently differently expressed that we could access in these samples. Maybe something unique we have in our cohort, these prodromal subjects. These subjects with an isolated REM sleep behavior disorder. Around 80 or 90% of the subjects who suffer from this IRBD, they will develop a disease in the course which is a synucleinopathy, so a disease where alpha-synuclein aggregates in your neurons. So these are patients at a really high risk. Jenny Hallquist, who's first author of the paper, she applied an algorithm to check what do these samples have in common, and if this algorithm learns to work with the data, we wanted to see is it enough to give this information to the algorithm to check if a patient is healthy or has Parkinson's disease or is in a prodromal stage.
They came to the result that the algorithm can identify Parkinson's disease subjects based on 8 of these proteins in 100% of the cases. Extraordinary. 8 of these proteins are enough to decide whether a patient is healthy or has signs of Parkinson's.
That's extraordinary. So then to elaborate on the link between REM sleep behavior disorder and the early detection or risk of developing Parkinson's, can it be considered an early stage symptom?
This is connected to electrical activity in the brain. It is known that there is a syndrome where people, when they're sleeping, they move a lot in these periods. Subjects that suffer from this, they have a high risk to get a neurodegenerative disease. Parkinson's disease starts at the brainstem and it distributes to the brain. And there is an area called locus coeruleus, so it's a blue area. And this has something to do with regulation of sleep and dreaming. And you can find pathology and changes in this area very early in Parkinson's disease. So the idea behind is maybe that these patients that have this sleep disorder, they have already early changes in these areas before the disease and the pathology spread through the brain and everywhere. And that's maybe the unique and specific part of our study, that we have a cohort of subjects with this, and that this sleep disorder is predictive, and this can precede these for years, 10 years, 15 years or something. So it's very, very early.
Michael's study mentions an enrichment in inflammatory pathways, and we're seeing a lot of that in many, many disease pathways, increased inflammation as one of the early signals. So what's the link between these pathways and Parkinson's in particular?
Inflammation plays a big role in neurodegeneration and in Parkinson's disease. We do not know if it maybe starts the disease or just triggers it, but it's present in these 8 ones. There's of the complement system, which is a part of the innate immune system. We found higher concentrations of proteins that are associated with the inflammation, but we also found proteins that are not so strongly connected, especially not in humans. So we have proteins of this WNT signaling, Wnt signaling pathway. These are proteins that are important for the survival of a cell. And from some animal models, we know that if these pathways do not work properly, then some of the dopaminergic cells, these specific cells of the PD subjects, can die. So we found some new proteins and we found some pathways that fit together with already known ones. We try to build up kind of a model where we show there are some protective mechanisms, there's some detrimental mechanisms in regard of the survival of these dopaminergic cells that are so strongly important to regulate movement in these subjects.
How does this compare to other well-known tools for diagnosing neurodegenerative diseases? And what are the next steps that validate it and establish it as a current diagnostic practice?
We have to connect our findings with all these established modalities that we understand. So it's important to see if maybe we see differences in concentrations of proteins in regard of the amount of neurodegeneration, the amount of dopaminergic cell loss, maybe in the imaging or something. So that's very important because then we can maybe build up some synergistic effects to see what do we already know. An advantage of our method is that the amount of blood that we needed is really, really small. It's about 30 microliters of blood for the test. We will have to see how our findings and where we can put them in the field and to see how they stick together with the already known approaches, imaging, MRI scans, and so on. With also, there is this seeding aggregation assays of alpha-synuclein and cerebrospinal fluid. This is something very new and important. You can diagnose the disease based on the CSF. It's more invasive, but we have to correlate our findings with all of this to see where we are.
In this episode, we examined advancements in Parkinson's disease research. Mariana Dichet's team discovered a novel genetic mutation that could be connected to a cause of early onset Parkinson's. Highlighting 2 sisters with juvenile Parkinsonism, the study linked their condition to a never-before-seen mutation in the SGIP1 gene.
This underscored a shift in Parkinson's diagnostics, moving us from focusing on neuron loss to examining synaptic dysfunctions which happen much, much earlier in the disease progression.
We also spoke with Dr. Michael Bartle, who discussed innovative approaches and biomarkers for early Parkinson's detection. Using proteome data and machine learning, Bartle's team can identify Parkinson's presence from only 8 proteins in the blood. This method is significant progress toward non-invasive early-stage diagnosis, which is pivotal in improving patient outcomes and enhancing clinical trial effectiveness.
These advances, while they still require further validation, they do promise a future where early intervention could significantly alter the trajectory of Parkinson's disease for patients around the world. This gives a new hope for people and the medical community alike.
Much like the hope provided by that very exciting case in Umeå that we started this episode with, where doctors used a magnetic camera-guided focused ultrasound to help people control shakiness in their extremities. So providing hope, providing earlier diagnosis is really important, and it's one of the great things that I learned recently. So since we started the show with Every Day is a School Day, let's take a moment and give a shout out to a wonderful organization making a difference.
I want you to know about Roald Dahl Marvelous Children's Charity. And it is marvelous. Their mission is to provide specialist nurses and support for seriously ill children living with complex, lifelong conditions. Their vision is that every seriously ill child has a Roald Dahl nurse specialist to help them lead a more marvelous life. And I have met some of these nurses, and they are indeed marvelous.
And back to the podcast.
And you can find a link to the Roald Dahl's Marvelous Children's Charity in our show notes. Thank you for listening to this episode of Discovery Matters. Our producer is Beth Armit Brewster, editing, mixing, and supervision by Banda Productions here in Sweden. Music from Epidemic Sound. My name is Dodi Axelsson.
And I'm Conor McKechnie. Make sure you rate us on Spotify or whichever platform you use to listen to our marvelous podcast. We will see you when we come back with another episode of Discovery Matters.